Hi all, I am Sach Fernando from Melbourne One TV, and today I have a very special guest, Dr. Danushka Wijesundara with me. Dr. Danushka is part of the world-leading University of Queensland team who are working towards finding a coronavirus vaccine. He's working under the guidance of Associate Professor Keith Chappell and Professor Paul Young, and also Professor Trent Munro, who are leading this vaccine project. How are you, Dr. Danushka? You doing
I'm
good?
well. Yes, I'm well. Thank you. Thanks
Thank,
for asking.
thank you, thank you very much for giving your precious time for us today. I know you're quite busy working on this very tough task. So, Danushka, can you please tell us about how you got to where you are today? A bit about your background.
Yes, so I was born in Sri Lanka.
Uh-huh.
I did a bit of schooling in Sri Lanka, but most of my schooling was done in Myanmar, a country neighboring Thailand. So I went to the International School of Yangon there.
Uh-huh.
And after I finished my schooling in Myanmar, I applied for university studies here
Yeah.
and got an opportunity to do undergraduate studies at the Australian National University with a bachelor of biotechnology.
Uh-huh.
And then I proceeded to pursue in my research interest in viral immunology, studying about viruses and also vaccine development.
Yeah.
So I did an honors and then did a PhD. After that, at the John Curtin School of Medical Research, and then went to do a postdoc, again pursuing my interest in vaccine development in HIV, hepatitis C virus, and Zika virus at the University of Adelaide, and then came to join the UQ team after that. So during that time, I've had great mentors. I think I should acknowledge them. Professor Arno Mulbacher, Dr. Matthias Regner, Dr. Muhammad Al-Sharifi, Associate Professor Sharani Ranasinghe, Professor Eric Downs. I think they've been fantastic mentors leading up to my position at UQ, now working with Associate Professor Keith Chappell, Paul Young, and Trent Munro.
Thank you very much, Danushka. So, I know we know that you are working towards a vaccine, working really hard with the University of Queensland research team. So, can you explain about the role of a vaccine for someone who doesn't know much about these medical terms, and how will a vaccine help a person to get immune to a disease?
Yes, so the objective of a vaccine is to actually introduce an agent, something that will safely train your immune system, your natural immune system, to actually recognize and respond against the actual infectious microorganism which you are vaccinating against.
Yeah.
So I guess that kind of answers the question. So it trains the immune system,
Yep.
and the immunity will remember. So it will stay there for a while, and when the real virus or real infectious agent comes your way, the immune system does not need to be trained. It's there to respond immediately against that infectious agent.
Okay, interesting. So what type of virus is COVID-19 or coronavirus?
Yes, so the coronavirus, the causative agent of the COVID-19 pandemic is referred to as the SARS coronavirus 2. So it is in the same family as the SARS coronavirus 1, which you probably know of. It caused the outbreak in 2002 to 2004, although it was much more restricted. It belongs to the same family. It's a beta coronavirus family member.
Okay.
And it also belongs to, there's also another member that's lethal in that family called MERS coronavirus, Middle Eastern Respiratory Syndrome coronavirus.
Yeah.
So it belongs to this family of coronavirus that are actually very lethal and can cause severe disease in infected people and deaths.
Yeah. And I think what the vaccine is trying to do is introduce, or perhaps to say provide herd immunity. to stop spreading the virus. Can you explain a bit about that?
Yeah, so it's not essentially there to purely there to satisfy the purposes of herd immunity.
Mm-hmm.
It is actually to protect anyone who gets vaccinated. And I guess the concept of herd immunity is that, if enough people get vaccinated, even those who cannot afford to get vaccinated, who are either immunocompromised or for some other reason cannot get vaccinated, can be protected, because the vaccinated individuals will not develop disease, and therefore they cannot transmit the disease to the individuals who cannot get vaccinated.
I see. So basically, like you're saying, the vaccine will prevent a person getting infected, as well as it will reduce the spread in the community. Therefore, it won't, go spread as fast as one would imagine.
Yes, that's pretty much, in a nutshell, that is what it is. Yeah, it can either prevent disease or keep the infectious agent to actually very low levels, that it won't result in transmission. So the community is protected.
I see. Yeah. Dr. Danushka, we heard last week the Prime Minister was saying the Australian government signed an agreement with the UK-based drug company AstraZeneca to secure a potential COVID-19 vaccine. I know they're still in phase 3 trials, and we don't know if it's going to get approved or not. And I know that you are working, and there's a lot of people around the world, I think around 160 vaccine candidates, both in preclinical and clinical trials. There are 29 right now undergoing clinical trials in humans. So how is your vaccine distinct from perhaps maybe to say the AstraZeneca, the Oxford? Now, when I read some of the articles published by UQ, University of Queensland, I saw that there's a special technology called molecular clamp, and it's patented by UQ. Can you please explain about this a little bit for us?
Yep. So I guess to answer your first question about how distinct our vaccine is, when I say our, it's actually a technology that was developed and patented by Associate Professor Keith Chappell, Dr. Daniel Watterson, and Professor Paul Young. It's actually Keith who originally came up with the concept of the molecular clamp. But how we're distinct from the Oxford vaccine, which is again a chimpanzee, it's actually a chimpanzee adenovirus vector vaccine. It is not what we use is a protein subunit vaccine platform. So basically, they have a virus which is carrying components of the coronavirus that will essentially, the virus vector they use is immunogenic. So it will train the immune system, again, to recognize the coronavirus genes.
I see.
And safely induce an immune response, or evoke an immune response that can neutralize the virus. Whereas our technology is essentially a protein platform. So we just take a piece of the virus.
I see.
So the piece of the virus that we take is called the spike protein. And we stabilize it using the molecular clamp technology. So when we stabilize, what it essentially does is the spike protein appears exactly as what you would get, or very similar to what you would get with the SARS coronavirus 2. So it's actually two different platforms in that regard. And that's essentially what the molecular clamp is meant to do. It's meant to take pieces of the virus that's on the surface that is crucial for the virus's life cycle or entry into host cells. And we take that piece and we train the immune system to recognize that piece of protein. And by doing so, we can make antibodies that can actually bind to the virus. When the virus infects you, it can bind to that protein and prevent the virus from infecting a cell.
Oh, great.
So therefore, you remain protected. So that's essentially what we're trying to do even with this vaccine, the SARS coronavirus 2 molecular clamp vaccine, which we call the S-clamp.
I see. This is very interesting. Yes, indeed. So is this something that's been done first, like the molecular clamp technology being used in humans? Like, has anyone else done this before?
So with the molecular clamp obviously being a patented technology, it's actually unique and novel to us at the University of Queensland. It's a novel technology for the whole world, but it's uniquely available to us.
I see.
And this will represent the phase 1 clinical trial that's ongoing for the SARS coronavirus 2. Will represent is our first phase 1 clinical trial with this technology. But we have used this technology against lots of other viruses. We have very promising preclinical data to show that this technology can work exactly as I described to you earlier. So yes, whilst it is the first in humans, I think there's potential that it can be applied for other diseases, viral diseases as well, going forward.
Yeah, so it's not limited to, let's say, just corona. So potentially in future, if there's another disease or a pandemic that could happen, there's potential that this vaccine can be used for that as well. Is that what you're saying?
Yes, absolutely. So when I, sorry, excuse me. So when I joined this lab, that was the intention, that we stress test this platform across random viruses, viruses where we don't have a vaccine for yet. There's very limited information available to develop a vaccine. So we're going to use this technology and try to make a vaccine for these random viruses. And therefore, when it comes to a virus from an unknown origin, that's actually causing a huge problem like SARS coronavirus to people, we can use it very quickly to make a vaccine and deploy it so that people can benefit.
Wow, that's certainly very promising. I think even the viewers would be really interested, because if we know there's a vaccine for a potential future disease, you know, even now it took nearly more than close to a year to get to this stage. So a future pandemic, I think it's probably better if there's a vaccine that could come into play sooner and would reduce the damage to the, you know, just for everyone. So right now, let's come back to your vaccine. So the vaccine and the trials that you're doing right now. So what's the progress with the UQ vaccine that you're working on right now?
Yes, so after receiving a lot of promising preclinical data, we have progressed our vaccine to phase 1 clinical trial. So we've shown that the vaccine can be effective and safe in clinical models. We're not taking any shortcuts in that department. So we've shown all those desired effects to progress in phase 1 clinical trials, and now it is being tested in phase 1 clinical trials. And it's progressing well. The outcomes of the study, the phase 1 study, will be available by the end of the year. And by that stage, I think you'll receive more definitive conclusions and data about how well our vaccine is performing in terms of primarily the safety. We don't want to harm anyone. Safety, and also with regards to the effectiveness in evoking an immune response, like neutralizing antibodies to neutralize the virus.
Wow, that's certainly good news for our viewers. So one thing that sort of came to my mind is that now we know viruses can mutate. How do you see now, is there a potential risk that this vaccine could be ineffective if the virus would decide to mutate itself? Or this vaccine is capable of even handling that?
Yes, so I think there is obviously a possibility that the vaccine could fail if the virus mutates quite dramatically, but I think all indications suggest that so far with the different isolates from different regions, the virus does not mutate so much with respect to the spike protein, which we're targeting.
I see.
But even if it did mutate, I think our vaccine is broadly targets lots of regions of the spike protein.
I see.
And therefore, even if there was a mutation in some region, I think the immune responses to other regions would compensate for that and hopefully help neutralize the virus.
Oh, wow.
So whilst it remains possible that the vaccine could fail, I think our early indications are promising that the vaccine and the immune response elicited following vaccination can counteract that effect.
Yeah. Wow, that's really good news. I mean, like you said, if it targets not just one part of the section of the virus, if it targets a lot of the protein sections, like you said, there is a high chance that even though the virus would mutate, the vaccine would still be effective. That is certainly good news for our viewers. Also, so I think moving on now, we know you're going with phase 1 and the results are still due to come. So if everything goes well and everything goes as planned, when do you think the University of Queensland vaccine will be ready, and who will be doing the production in Australia? Do you have someone already in place to make productions available?
Yes, so the production will be handled, so provided the phase 1 clinical trial goes according to plan and it's safe and immunogenic.
Yeah.
Our industry partner in Melbourne, the Commonwealth Serum Laboratories, or CSL, they will look after the large-scale manufacturing of the vaccine so that it can be available to the wider community, Australia and elsewhere.
Oh, wow.
So at this stage, I don't want to make any promises. I guess we still have to go through the phase 1 clinical trial and see what the results of that. And if, I mean, I guess the objective, and I think there's a press release by CSL, our industry partner, who said that their objective is to make millions of doses widely available by the end of next year. So at this stage, we're progressing at pace to actually meet that target.
Yeah. Wow, that's certainly good news, and obviously fingers crossed. Everyone's wanting to get out. We are from Melbourne, so we're still in lockdown, and we can't really get out. I know you are in Queensland right now. How's Queensland going? Is it restrictions it eased now and moving?
The restrictions I should say more ease than what you get in Melbourne, obviously. But we do have these occasional incidences, and that's actually a bit dangerous because it does suggest that it hasn't gone away. And I think hopefully it will not become the situation that is in Melbourne.
Yes, we certainly hope so.
But there are certainly elements against complacency. There's information against complacency that we should not just go back to our normal lives.
That is
just
true.
Because Brisbane seems to be doing much better than other places, or Queensland's doing much better than other places. It's still not gone here.
Yeah, that's true. That's true. Thanks a lot, Doctor. I think we covered a lot for our viewers, and we did talk about the vaccine and the progress of your vaccine that you are working right now. We really appreciate your time that you gave us today, and all the best. And we wish you luck with the future ventures you are about to do, and especially with the vaccine. I think everyone will be quite keen to see what happens closer towards December. All the very best, and we wish you success. And thanks again. And I would like to sign off from Melbourne One TV. Would like to come back again with another video. Give us a thumbs up and subscribe. And thanks a lot. Bye-bye.